goal for the day is to explain how organisms get energy when there isn't oxygen. So when it comes to like organisms that are within systems underwater or just in an area with little to no oxygen, they still need to be able to get the energy they need, or break down sugars or break down energy sources and get what they need out of it. So there is kind of a backdoor kind of way to do this process without oxygen. Okay, so what is fermentation? That is not very big font because I don't know why. All right, so there we go. So in the absence of oxygen, fermentation releases energy from food molecules by producing ATP. Just like cellular respiration does with energy, fermentation does it without oxygen. This includes glycolysis. Remember, glycolysis doesn't need oxygen. But what's different between cellular respiration and fermentation is what happens after glycolysis. So we know that when there's oxygen available, we will go straight from glycolysis to the Krebs cycle, and from the Krebs cycle to the electron transport chain. But some organisms can't handle high oxygen levels. Some organisms don't have access to high oxygen levels. So they had to figure out a way to get from glycolysis to the energy they need without oxygen available. So this includes glycolysis and some other process that doesn't involve an electron transport chain. There's actually two of the pathways that we're going to talk about. So no oxygen, fermentation. This means it is anaerobic. We talked about how if a pathway needs oxygen, it is aerobic, and if a pathway doesn't need oxygen, it is anaerobic. So fermentation is an anaerobic process that gets all of the energy from food molecules, getting ATP out of it, and doesn't have oxygen readily available. So, actually a really cool process. All right, let's keep down. All right. So, what happens during fermentation? So, if we remember glycolysis, I'm going to write this out over here so I would have some sort of doodle. We start with glucose, right? That's glycolysis. It's our 6 carbon sugar, and we're going to get from our 6 carbon sugar to our 2, 3 carbon sugars. And when that happens, we use, or we get to ATP, right? Or actually, yeah. We use 2 ATP and get to a DP, and then we take 4 ADP in a later step, and we get 4 ATP, so we have a net of 2 ATP. So we've got some ATP already. But we also have high energy electrons. And those high energy electrons, like you diagrammed yesterday, usually go straight to the electron transport chain. You're going to get 2 NAD pluses, and we're going to take the electrons off of that glucose, and we're going to get 2 NADHs. That's our high energy electron carrier. So that's what happens during glycolysis. In fermentation, glycolysis still happens. We still have this process taking place, but we need to replenish this. So in the electron transport chain, we're going to transfer those electrons to the electron transport chain, and we're going to get this NAD plus back. And that NAD plus will go right back up to glycolysis and get recharged. So cells will convert NADH to NAD plus by passing high energy electrons back to pyruvic acid at the end product. So what will happen is these pyruvic acids and these NAD pluses will give us NADHs and some other 3-carbon process, if that makes sense. Because we don't have, actually, that's really confusing. We don't have the NAD plus to go back into the cycle to continue glycolysis if there's no way to replenish it. The whole goal of fermentation is to replenish this NAD plus so glycolysis can continue. Because if we don't have an electron transport chain, there's nothing to get us from NADH back to NAD plus. So, there are 2 ways this can happen. Lactic acid fermentation, which is what we can do in dire circumstances. And there's alcoholic fermentation. We can't do alcoholic fermentation in our cells, but we use a lot of cells that do. So we've got lactic acid fermentation and alcoholic fermentation. Both of those are different pathways to get NADH back to NAD plus so we can continue glycolysis. So 2 types of fermentation: lactic acid fermentation, which is what we can do, and alcoholic fermentation, which is what we utilize in other organisms but can't necessarily do ourselves. And these will pop up as section titles, so if you don't get them, they'll come back. The first one is alcoholic fermentation, where we take pyruvic acid and NADH, we add that NADH to the pyruvate, and we get alcohol, carbon dioxide, and NAD+. And that NAD+ will go back to the beginning of glycolysis. So, glucose into pyruvate. This part is exactly what I have on the board right now. Then the NADH from glycolysis will go back to pyruvate. And when it goes back to pyruvate, we're going to lose a carbon and we're going to get ethanol. If I can have your attention over here, please. Alcoholic fermentation isn't done by us, but it is done by yeast cells. So is this the worm? Yes. So we are going to see this kind of process take place, and we're going to see the production of carbon dioxide. So I'm just going to put some warm water because enzymes like warm. This is just regular table sugar because the yeast needs something to eat, our glucose. And these are just dried out versions of bacteria, which is yeast, which is something we use in a lot of like food substances. So I'm going to mix that up really quick, and then I'm going to starve it of oxygen. So by covering the top of this, oxygen can't get in there. We're going to watch as those yeasts start to ferment, as these yeast cells start to break down that sugar, they're going to release carbon dioxide. So we're going to let this sit and we'll come back to it in a moment. So alcoholic fermentation. Yeast and some other microorganisms can carry out this process. It produces alcohol and carbon dioxide. This is the process that makes alcoholic beverages and makes bread dough rise. So as these yeasts break down the sugars that are naturally put in there, because I chunked a bunch of sugar in there, they will break down that sugar and release carbon dioxide. This is what makes little pockets in bread dough. Because if we put bread dough, we put yeast in it, we put sugar in it, we knead it all up, and we put it someplace warm to do its thing. As those yeast cells start to break down the sugars that we put into bread, they'll release carbon dioxide and it'll puff up. That's the whole point of like proofing dough. As that dough puffs up, as that carbon dioxide is released, we get these pockets, which is why bread is soft and fluffy and not super dense. In fact, the breads that don't use active dry yeast will use like baking soda to try to do the same thing, but they're usually not as soft or fluffy as the rising flours, the rising doughs. This is also very similar to how we make alcoholic beverages, beers, and wines. All use yeast to break down sugar and release alcohol. But then we use the alcohol instead of the bubbles, which is also why like a glass of beer bubbles, why champagne bubbles, is because it has that carbon dioxide that is released by the yeast as they break down sugars. Make sense? Cool. It's actually really versatile, and we use it in a lot more stuff. But then there's lactic acid fermentation. Lactic acid fermentation is a little less complicated than alcoholic fermentation because we aren't losing carbon dioxide. We only have one product. What's going to happen is we are going to do glycolysis just the same glycolysis process, but we're going to take that NADH, we're going to add it directly to that pyruvate, and it's going to give us lactate or lactic acid. We're just moving some atoms around. So instead of adding those electrons and breaking that pyruvate, we're going to add those electrons and change that pyruvate into lactic acid. So in both scenarios, in alcoholic fermentation and lactic acid fermentation, the whole goal is to take this NADH and get rid of it. Give us NAD plus back so we can keep doing glycolysis. Because if we don't have NAD plus, we can't break down glucose anymore because there's nothing to take those electrons. So in alcoholic fermentation, we take sugars, we break it down, we use the electrons from NADH, and we make ethanol or alcohol and carbon dioxide. In lactic acid fermentation, we're taking sugars, breaking it down into pyruvic acid, using those electron carriers, adding those electrons straight back onto pyruvate, and we get lactate or lactic acid. So why do we use lactic acid fermentation? A lot of the same things, or not same things, but we still use them for food culture-y stuff. We use bacteria that do lactic acid fermentation to make cheese, yogurt, buttermilk, sour cream. That like kind of tangy taste that comes with sour cream, and you get a little bit of it in yogurt, is because of this lactic acid. As these bacteria break down the sugars that are naturally in milk, and they produce lactic acid, we will get cheeses, yogurt, buttermilk, sour cream. The pickling process also has to do with fermentation. So we put the natural bacteria that are on the surface of our vegetables or whatever we're pickling and use those to kind of break down sugars and create lactic acid. And that lactic acid is something that we think tastes good, or some people think tastes good. I'm not a big fan of pickles. So humans, our bodies, can do this process too. In extreme cases, not for long term, but for super short term, if we don't get the oxygen we need, our muscle cells can do this process. So, exercise. Hey, look, our balloon stood up. So, our body produces ATP differently in different stages of exercise, right? There are two main sources of ATP. ATP that's already in cells that exist from cell respiration, and ATP that is from fermentation. So we're going to use muscle cells in this example. So whenever we exert ourselves, whenever we run, whenever we do like hardcore physical exercise, we need to breathe more, right? And we need to breathe more because our electron transport chain needs oxygen. But if we can't get enough oxygen into our bloodstream, if we can't get enough oxygen to our muscle cells, but they still need the ATP to run, they'll start fermenting. They'll start taking those NADHs from glycolysis, adding them to pyruvate instead of sending them through the Krebs cycle, and using that to get ATP for super short term. Make sure this balloon won't pop off here. So in our example for human cells, we're going to specifically look at muscle cells because they are the ones that do this the most often. Okay, so when your body needs energy in a hurry, like we're talking fight or flight responses, we're talking like sprinters. When your body needs a lot of energy really, really quickly, like for some reason you just take off. For quick bursts of energy, we will use up all the ATP that already existed, all the ATP that was around from cellular respiration. And then we'll start doing lactic acid fermentation. Your muscle cells will start producing lactic acid, and that's why our muscles burn. You know how if you do some sort of quick exercise and all of a sudden your calves just hurt, like you cannot run anymore because it hurts too bad? Like the very, very end of a sprint. And I don't know, I don't do track, but like when I was in high school gym, right? You just run so hard that your muscles start to ache and you cannot run anymore. It is because those muscles don't have enough oxygen and they are producing lactic acid. This is why you rub those muscles after you stop, when you stop, because we're going to try to spread out that lactic acid and break it down. So when we are doing quick bursts of energy, our bodies will start fermenting instead of sending things through cell respiration because we don't have the oxygen that we need. And it hurts. That's why we can't do it for very long. That's why it is short term. Doing okay? Does anybody have any questions about this? I think it's super cool. All right. Leave. So that's short-term energy, but whenever humans run, right, it's not just about getting from point A to point B from in one it getting from point A to point B as fast as possible. That was the sentence I was trying to say. What about from marathon runners, cross country runners, people who do exercise for a very prolonged period of time. The only way to get this energy long term is through cellular respiration. For longer exercise, cellular respiration is the only way to get our supply of ATP, because cellular respiration releases more energy, but it's also slower than fermentation, which is why for long-term exercise, you need to keep up a certain pace, right? You can't like when you sprint, you can go a heck of a lot faster than you can if you're running long distances, but that short distance burst of energy isn't sustainable. You can't go the same pace in a sprint as you can in a marathon. And that's because there's not enough oxygen getting to your muscles for them to do cellular respiration and get you the energy you need to do the speed of a short distance long term. This is why we use exercise to burn fats, right? When you burn through all of the sugars and all of the ATP, your body starts breaking other stuff down. So sugars are fast, sugars are easy. That's why sugar rush exists, right? Sugars are easy to break down. But when there's no sugars available, none of those quick carbohydrates available, when you run through all the sugars, when you run through all the ATP, you start to break down other molecules for energy, proteins and fats. This is why marathoners carbo load before races, because carbohydrates are energy that they can break down quickly. They'll have a bigger store of these before their body starts breaking down proteins and fats. And that's lactic acid fermentation. Does anybody have any questions? I'm going to come back to our little balloon thing because it is clearly bubbling up. So, as you can see, not only did our balloon blow up, right? We've also got all of these bubbles. So all those yeast cells have frothed up because they're creating carbon dioxide. As those yeasts, there's no oxygen available in here because we've got our balloon covered. It's not exposed to the open air. Because our yeast cells are exposed or not exposed to oxygen, because they have to carry out fermentation, what they're going to do is as they break down sugars, they're going to release carbon dioxide and alcohol. So all of these bubbles would be essentially like if you have a glass of milk and you put a straw into it and you blow it up and it starts fluffing up, except instead of your breath, it's the carbon dioxide that is being released by these yeast cells. So the gas in this balloon is the carbon from the sugar that I put in there. As they broke down the sugar, they released the carbons that were in here, and they are now in a gas form in carbon dioxide. Super stinking cool. Catch my microphone on something. Yeah. So that's all I have for you for fermentation. Your exit ticket is a little bit longer and requires some effort and imagination. So please open up your Chromebooks and do the exit ticket for the day.